Today I'm going to talk to you about the development of potential new agents for treatment-resistant depression. So as many people would know, this is a disease that needs no introduction, that's fairly common in society with a large population of people who are refractory to the treatments that we have. And just in the recent decade, there's been a very large success with esketamine, which has reached over $1 billion a year in revenue. However, this is limited by the fact that it's essentially dissociative, has to be given in a REMS supervised setting. And then even more recently, many people will have heard about psychedelic drugs, which have shown a lot of promise for this disorder. They have a very substantial efficacy in the treatment of depression and treatment-resistant depression. However, there are a few problems which limit their widespread use. Number one, because they're hallucinogenic, that creates a huge problem for placebo response. We really don't know if they're better than placebo because there is a fundamental issue with a drug that has such a strong subjective effect. Secondly, because the drug is hallucinogenic, it has to be given in a highly supervised setting over a long period of time. So because of this, there's been a huge interest in the development of non-hallucinogenic psychedelic drugs. Just in the past few years, there have been some very major acquisitions of products that are aiming to fill this need by providing non-hallucinogenic psychedelic drugs. That is a drug that would have this long-standing efficacy for depression, but without the huge subjective hallucinogenic effects. However, all of those ones that I showed you on the prior slide are drugs that are pushing at the serotonin 2A receptor. This is the common receptor that's involved in the action of these drugs. However, there's an emerging literature that's made it clear that these drugs activate a panoply of related monoamine receptors that go far beyond the serotonin 2A receptor. And in fact, this may be responsible both for their therapeutic effects as well as they may be able to counteract the hallucinogenic effects of these drugs. And this is based on this Neuron paper that some people may find interesting. So rather than adjusting for creating a biased agonist at the serotonin 2A receptor, our strategy is to activate the serotonin 2A and another receptor. And in this paper, which is currently in press, we showed that in a mouse proxy of the hallucinogenic response called the head twitch, this activation of the alpha-2 adrenergic receptor counteracts this hallucinogenic effect of psychedelics. And while it does that, it also preserves the antidepressant effect. This was quite robust across different classes of psychedelic drugs, and there was no change in the antidepressant effect in this combination treatment. To develop this further, we filed a patent application for combination therapy. But in this proposal, what we're hoping to do is to develop a new small molecule. And looking at the pre-existing data set of the polypharmacology of the serotonin 2A agonist, we found that there was a potential target for a drug that might have alpha-2 agonist activity and serotonin 2A activity. So this would be a drug that activates both receptors, thus counteracting its own hallucinogenic effect. There are also planned human studies to de-risk using the combination therapies this novel compound. So we're going to proceed along two aims. One is to look at large library and develop a large number of synthesized compounds, which we can test in vivo in preclinical model. And secondly, using our literature-based lead, we're going to develop, using molecular dynamics simulation, a smaller number of compounds which we think can be tested in animals as well. Our team includes experts in translational psychiatry and clinical trials, including John Crystal, who helped to develop ketamine as a treatment for depression, as well as Carl Sumavori, who's an expert in computational pharmacology and GPCRs, including the serotonin 2A receptor. We also have capabilities to conduct more advanced plasticity assays. So after the development of this compound, the next phase of our program, which is also supported by NIH as well as Department of Defense awards, will be to do a more thorough characterization of the plasticity enhancing and behavioral change effects of these drugs to therefore enable IND and human studies. So in this first phase, we will then develop the small molecule according to the two platforms that we discussed and test them in animals. So with that, I'm eager to answer any questions that people may have.
So you haven't found the molecule yet. That's the plan. So obviously the issue of psychedelics has always been not a composition matter of method of use patent, and the way you're overcoming this is to find. So can you go back to slide 6, because that was like a really important slide that we didn't spend more than a couple. Yeah. So
The plan, yeah.
yeah. So you haven't found the compound yet, but this is your strategy for. Okay.
So, but I will say, so we already have the patent application for the combination of drugs that can activate the alpha-2 receptor and the serotonin 2A receptor. So this new composition of matter intellectual property that we would be pursuing with this would layer on that.
But the current IP is method of use, right?
Yes.
So according to that slide, are you looking at 30K to go from hit identification to in vivo POC?
Yes. I mean, as I mentioned, this is, I think so, because for one reason, the preclinical models are very affordable and are already going within our research group, and the work is also supported by NIH as well as the DoD funding in the psychedelic area.
Right, but you have to identify potent biselective compound with good PK and brain penetration to get into that model.
Based on the literature that we mentioned, there are already compounds which activate both receptors and have subjective effects in people. What we believe is that what is off about them is the ratio of the alpha-2 receptor activity versus the serotonin 2A activity. So it's more of a fine-tuning an existing target.
Okay, thanks.
All right, if there's no other questions, please thanks Alfred. So I see a lot of people in the audience